This video is a brief overview of the different types of passive transport. Now, before we dive into the three different types, I want to review the two big features of passive transport. So remember, the first one, it does not require an input of energy. Okay, and I apologize, I'm writing with my mouse again, so it's looks like a second grader wrote it, but that's okay. So no additional input of energy is needed. This happens all on its own. Okay, it happens passively, naturally, hence the name passive transport. The other feature of passive transport is that molecules move down their concentration gradient. Okay, now I'm going to do shorthand because, again, this is hard to write with my pen. So down their concentration gradient. And so I just want to remind you that in science, these little square brackets that I have on the other side of gradient, that's the scientific indication or symbol for concentration. So when I say things are moving down their concentration gradient, that means that molecules are moving from an area where there's a lot of those molecules to an area where there's a little bit of those molecules, okay? So the first type of passive transport is simple diffusion, okay? Now the picture on the right, there's just kind of a lot happening. So I'm gonna kind of sketch a different picture here. So we're gonna pretend that this line here is my plasma membrane. Okay, it's that phospholipid bilayer in their picture. So I'm just gonna abbreviate it PM for plasma membrane. And essentially with simple diffusion, what that picture is trying to show is that molecules move from where there's a lot. Okay, so let's say that this is the starting situation, okay? So there's a high concentration. So again, these square brackets mean concentration. So there's a lot up on the top of the membrane, okay, outside of the cell, and there's fewer, right, a lower concentration on the inside of the cell, this bottom region. So what's gonna happen is molecules are going to naturally cross this membrane. Okay, so they're moving from where there's a lot to a little. In other words, they're going down their concentration gradient. And this is going to happen, okay? So pretend what I'm drawing now is later. Eventually what happens is there's going to be the same amount on both sides. Okay, they kind of want to just balance each other out. So that's what's happening in this top picture, okay? So on the left here, that's supposed to be an A. That's like the starting situation. Molecules are naturally crossing the membrane over time. So they're moving again from high amounts up at the top to low amounts in the bottom. And then in that third pane, if you were to count them, you would see that there is about the same number on each side. Maybe there's the exact number, I haven't counted. Okay, but that's what happens. But I want you to notice here, and I'm sorry, I've taken up, let me see if I can erase some of this. What I do want you to notice, though, is that these molecules are moving across the membrane on their own. There is no protein helping them, okay? No protein helping, okay? These molecules are able to cross the membrane all on their own. So there's an important feature of these molecules that are able to essentially just wiggle their way across the membrane. Okay, so these molecules are going to be small, non-polar molecules. Okay, that means they're typically hydrophobic and they are not charged. Okay, in other words, they're more compatible with that membrane because they are able to cross that hydrophobic barrier on their own. All right, so now let's go down to facilitated diffusion. So notice, y'all, that diffusion is in the title for both of these, right? So that tells you that the overall process is the same. Okay, molecules are still going from high amounts to low amounts. They still do not need any input of energy, okay? But this time, they do need some help. That's what facilitated means. To facilitate means to help. So if you look at this picture down there, do you see, I'm gonna draw an arrow to it, this transmembrane protein. Remember, transmembrane proteins are big enough that they cross the entire membrane and they're hollow inside. And so if you look at this picture, okay, these green molecules are moving down the membrane. I'm sorry, my dog decided to just start playing with this one. Hi. Sorry about that. So these green molecules, right, there's a lot outside of the membrane or on the top of the picture, and there's fewer inside, right? So they are going to go down their concentration gradient. So these green molecules are moving down, okay, with the help of this transmembrane protein. But now let's look at the red molecules. Okay, so they're also going, I guess this one's not showing that picture. So they're also going to go down their membrane. So notice again, there's more red ovals at the top or outside of the cell. And they too are going to go down this protein or through this protein to go inside of the cell. Okay. So the only difference between facilitated diffusion and simple diffusion is that facilitated diffusion uses a transport protein. Okay, it uses that transport protein. And remember, that protein must be a transmembrane protein. It has to span the entire membrane and be hollow. Okay, but remember, transmembrane proteins are a type of integral protein because see that helper protein right there, it is literally embedded into the membrane, right? It's stuck in there so that it can't move and disappear. And it's hollow so that things can pass through, okay? Now, our last type of passive transport is also a type of diffusion, but it is the diffusion of water, okay? And there's a pretty picture on the last slide, but I'm going to sketch it out while I talk because it's just easier that way. So osmosis is the diffusion of water. So it's going to have those same features, okay? No extra energy required. And these molecules are going to move from high amounts of free water to low amounts of free water. Okay, now you might be asking yourself, what is free water versus non-free water? It's kind of a weird thing to say. But what it's getting at is that water is the universal solvent. It loves to work and dissolve things. That is its jam, okay? Work, 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 right? Friends with Rihanna. Anyways, sorry, I crack myself up sometimes. Okay, so water wants to work. It's going to go where the solute is. So if there's lots of salt outside the cell, that's where water wants to go, okay? If there's lots of stuff inside the cell, that's where water wants to go. So free water just means water that isn't working, right? So if I'm off the clock and I'm sitting on my couch, you know, binge watching Downton Abbey, which is what I've been watching lately, right? I am free to go do other things. I am not working. Okay, so free water is water that is not actively like dissolving solute. Okay, so that means water wants to go where there's work. So there are three situations you need to be familiar with. So first things first, let me get this pen working. Here we go. One situation, that's my cell, is there's a lot of solute. Let's just use sugar. I like sugar. Just had a lot of it in my coffee. Pretend there's lots of solute outside the cell and there's a little bit inside the cell, okay? So the solution, that which is outside of the cell, has more, okay? So we call this a hypertonic solution. That's a T, hypertonic solution, okay? So again, if you're hyper, what does that mean? It means you have a lot more energy than usual. So a hypertonic solution means the solution, the stuff outside the cell, has more solute than what's inside the cell. Okay, so again, hyper means more. So in this case, remember I told you that water wants to go where there's work that can be done. So where is there more solute, more things that can be dissolved? That's outside the cell, right? So in this case, water is going to want to rush out of the cell, okay? So this results in a situation called plasmolysis. I'm gonna get my pen from my office soon so I don't have to keep writing like this. Plasmolysis, and that's where the plasma membrane of the bacterial cell kind of shrivels. It detaches from the cell wall, which is something that a structure we're gonna talk about in a few minutes. But essentially the plasma membrane starts to detach from this outer cell wall. So it looks like it's shriveling, but you don't use the term shriveling when you talk about bacteria, which we call it plasmolysis, okay? All right, on the other extreme end, my pen keeps going away. I don't know why. Okay, on the other end, opposite situation, perhaps we have more sugar inside the cell than outside the cell, okay? We call this a hypotonic, hypo and then tonic, so sorry about a space, hypotonic solution. And so in this case, again, water always wants to go where there's things to dissolve. So there's more stuff inside the cell. So the water is going to rush into, should be an artist really. Water is going to rush into this cell, okay? And we're going to say that the consequence of this is that the bacterial cell will swell. We don't say burst because as you'll learn in a future video, the bacteria have these really strong cell walls. So it takes a lot for them to actually burst. It's not impossible, but for right now, we're just going to use the term swell because that always happens, okay? The bacterial cell, we say that they will swell because water is rushing into the cell, okay? So again, compared to the outside solution, there's more stuff inside the cell, there's less in the solution. So when there's less solute in the solution, we say it is a hypotonic solution. All right, and then in the middle here, we have the situation where there's the same amount of solute inside the cell as outside of the cell. This is called an isotonic solution. Sorry, that's a C, isotonic solution. Wow, handwriting's getting worse. And so in this case, because there's the same amount of solute outside the cell as there is inside the cell, there's not going to be any rushing of water, okay? Water is going to be entering and exiting the cell at the same rate. So there's no change in the cell appearance, okay? All right, that's the end of this video. Please note that there is another slide, I guess I can show it real quick, that shows you what I just said, okay? So I do want to point out though over here on the hypotonic solution, notice it says the cell expands and can eventually lyse. So if the solution is hypotonic enough, then enough water can rush in and it can lyse. Lyse is the science term for burst, okay? It explodes, so to speak. But again, if I were to ask you about this on an exam or a quiz, we're going to go for now with swelling. All right, y'all. Have a good day.